Good afternoon everyone and welcome to our today's online lecture, the second section of our mutation that we discuss in our other lectures. Now to bring your attention here, mutation, we know that it is a change of nucleotide sequences that change the gene normal sequence, and it can lead to very dangerous situations, and sometimes it can be beneficial to the organisms. What we're going to talk about today basically is the major types of mutations that we normally see. And to begin with, mutation being heritable means that mutation, if it occurs in one organism, it can be transferred to the other generation. It has a heritable genetic character. So it's something that can be passed from generation to generation in a very slow rate. So we also have a mutation sometimes can be very silent. What that means is if these nucleotide substitutions occur, normally it does not change the type of amino acids that are produced by that particular gene region. In most of the cases, it happens in the third codon. So third codon mutation normally is a silent mutation. If it happens, it does not change the type of amino acids that normally produce. And we'll give you that example in a short while. But to understand that codons are 3 nucleotide base pairs. So if that third nucleotide is changed or substituted by any other nucleotides, normally did not have any negative impact or any positive impact of the gene. The products still remain the same. So macro alteration and minor alterations are terms that we normally use in describing some concepts in mutations. So the macro alterations are when there is a very large changes of a particular gene region, either being substituted or duplicated or deleted or inverted, or maybe it can even rearranged. We call that a macro alterations. That is, it must involve a very large region. For example, let's talk about 50 nucleotides. 50 nucleotides. Now, if these 50 nucleotides is duplicated twice or 3 times, that is a huge duplication. So that would be a macro alterations. But in the macro alternation, that is only when a single nucleotide base is either deleted or duplicated or inverted. So the macro involves a very large section of a gene being mutated, and micro becomes a very small, in other words, to be specific, a single base pair becomes deleted or inverted or duplicated. So the basic types of mutations that are common are transitions. So transitions, to be more specific, involves a changes of nucleotides from one region to the other. Or in other words, the purine changes to an alternate purine. So like purine base will be substituted by a purine base. So the pyrimidine also can be substituted by the other pyrimidine nucleotides or analog. Let's take a look at this. For example, adenine can be substituted by thymine, or in the case of the other one, cytosine can be substituted by guanine. So that is the transition, and normally this is the likeliest chances of a mutation. Mutation is more common in transition than transversion because of they being the same pyrimidine or purine. So transversion is the opposite, when a pyrimidine is substituted by a purine, or a purine is substituted by a pyrimidine. For example, adenine can be substituted by cytosine, or G can be substituted by thymine. So it is very possible for all these 2 circumstances or cases to occur. And when we look at the reason why transitions are more common than the transversion is because of the size of the bases. The other ones are more common in our genes than the other ones, and this is one of the reasons. So in our subsequent lectures, we are going to look at mutation in details. But until then, stay tuned and enjoy the video. Any question or doubt, just notify me via my regular email address or my YouTube.